I'm excited to now welcome Dr. Adam Boyco to the stage. Dr. Boyco is Embark's founder and chief science officer. He is a graduate of the University of Illinois, Urbana-Champaign, and received a Ph.D. in biology from Purdue University before his postdoctoral work at Cornell University and Stanford University. Dr. Boyco is currently an associate professor in biomedical sciences at the Cornell University College of Veterinary Medicine, focused on the genomic investigation of dogs. Dr. Boyco's research has addressed some of the fundamental questions about canine evolution and history, and his lab has uncovered the genetic basis for many canine diseases and traits. He has traveled the world collecting dog DNA samples and has contributed to numerous scientific studies. His work has helped inform some of the genetic health tests relied upon today by breeders and dog owners. He is an expert in numerous canine health and genetics topics and will answer questions live after his talk during the Q&A portion. Please join me in welcoming Dr. Adam Boyco.
Hi, welcome to the Embark Canine Health Summit. I'm Adam Boyco, Associate Professor at Cornell University and also co-founder and Chief Science Officer of Embark Veterinary. Today I want to talk about genetic testing and the opportunity to drive scientific discovery and improve the future of dog health and longevity. And this plays near and dear to the research that my lab and other labs do, and that Embark does, and the reasons why we founded Embark in the first place and are using it as an engine of discovery to help improve dog health. I like to start my presentations, I just downloaded this morning from OMIA, the Online Mendelian Inheritance in Animals Atlas. There's a lot known about dog genetics. Dogs are a very well-studied species, and when you compare across all the other species that geneticists look at, more is known about the genetic basis of canine traits and disorders than any of the other species in this database. So if you're not a human or a mouse, being a dog, we can say the most about you based on your genome, at least when it comes to large effect mutations that influence various disorders as well as different traits that have been selected for. So we're up to over 300 known causal mutations that have been identified in dogs, and most of which are available through genetic testing from various providers, including panel testing from Embark. My lab at Cornell started 11 years ago. We were really focused on trying to identify using large data sets, whether there's any other conditions that we can identify causal variants for. The traditional approach to doing this is the Genome-Wide association study, and so my lab was doing some of the largest studies in dogs at the time. And so for this study, we looked at 123 different breeds of dogs and genotyped over 1,500 dogs on what was at the time the densest genotyping array, 180,000 markers. And we asked pretty simple questions about body size, about coat color, and in this case, we were looking at fur shedding. And we weren't measuring the fur shedding in individual dogs, although we joked about, trying to do that either with like sticky gloves or vacuum cleaner, the different ways that you could try to do that. But instead, we just looked online and how the different breeds were reported for fur shedding. So it was really the simplest kind of study you could do. And yet, because we had all of this data, we were able to get really clean associations. First with the R-spondin2 locus, which we already knew about from work out of Elaine Ostrander's lab over 10 years ago. We know that that's the gene that underlies the wiry coats, the poodly coats, the low shedding coats for the dogs that carry that variant. But we also identified this other previously unknown association on chromosome 1 at this melanocortin 5 receptor. And so Jess Hayward, who was in my lab at the time, did a lot of digging to understand the genetic interactions going on here. And obviously, carriers of this R-spondin2 are going to have a low shedding coat. But if you don't have that homozygous poodle coat, then the difference between kind of being a low to medium shedding dog versus a high shedding dog is this melanocortin-5 receptor locus, and it's a missense mutation in the gene that we believe underlies the phenotype, and sort of the difference between this highly shedding, Siberian Husky or wolf coat versus this lower shedding and shorter, pug and boxer type coat. And so applying big data, even with very simple kind of phenotyping, has this ability to identify new traits in dogs, and so that had been the focus of a lot of research. But even though we found all of these disease associations and trait associations, there was still a lot that at least I felt like still needed to be done in dogs, and like really basic questions like, well, most dogs eventually develop a complex disease, something like cancer, an autoimmune disease, orthopedic disease, cardiovascular disease. The genomics behind those have been much harder to try to disentangle. And we know from human studies, it takes, not 1,500 samples, it takes 15,000 samples, 150,000 samples. It takes large data sets in order to start to disentangle the genetics of these. We know behavior has also been more difficult to disentangle. Different breeds behave different ways, and within a breed, there's a lot of variation in behavior. Whereas the genes for body size are well known, the genes for behavior are not well known. And again, this is a question that we think is going to require larger data sets. And then also the genes for longevity. I mean, dogs are a really interesting organism for thinking about longevity, because within one species, you could have a twofold difference in aging rate, as well as a difference in developmental rate. So what are the genetics underlying that? We know size plays a large role, but we don't know, do all size genes contribute to longevity equally, or are there other modifying genes that are important, and why do you have this relationship in the first place when it's the opposite of what you see when you look across species? And I think everybody, wishes dogs live longer. Like, why do cats get to live longer than dogs? It doesn't seem very fair at all. So these are all, areas that are of intense interest to me and to other researchers in the field. And in order to really make progress here, we need large-scale studies that have hundreds of thousands of dogs. 5 or 10 years ago, thinking about this, we did have large-scale studies, but they were in humans, and you had this model of these consortia projects that were largely, funded through, research organizations, governments. The Wellcome Trust Case Control Study being one of the pioneering studies, but lots of other, studies there that were looking at 100,000 or more individuals and doing these dense genotyping studies so that you can make these associations. A lot of great research has come out of that. Unfortunately, the model of funding academic research for animals doesn't really support, getting National Institutes of Health funding for this kind of study. I mean, I think the exception to the rule is probably studies like the Golden Retriever Lifetime Study, which was funded through private donations of over $25 million, and still that's a study with 3,000 breeds. And so we know we want much larger scale studies. And the model we looked at that we saw potential in was this model of genetic testing, personalized genomics, really. And if you look at the human side, and companies like 23andMe and Ancestry DNA, they were amassing data sets with tens of thousands of people. And dogs are part of our family, right? So people were paying for genetic testing in dogs, but they weren't doing it with these dense genotyping arrays where you could actually use the data to drive discovery. And so, looking at the landscape in 2015, when my brother and I were thinking about how do we want to expand on this research endeavor, there was sort of this blank space where, yes, there was dog DNA testing, but nobody was doing it on research grade arrays. Nobody was taking that data and trying to take that data to the next level of building data sets to ask questions and answer questions about cancer and hip dysplasia and allergies and things like that. And so we saw an opportunity to do that, and so we founded Embark in 2015. It was in the incubator at Cornell, the life sciences incubator, until it graduated, and now we're headquartered both in Boston and having a research core in Ithaca. And we launched the consumer test in 2016. So we've been testing dogs now for over 5 years. with very much of a 23andMe kind of style, where you're using a research array and you're also asking customers to answer questions about their dogs, so that you can make these kinds of associations. And I'm pleased to say customers love doing that. We have lots of customers coming back to the website every single month, looking for new updates, answering new research surveys, and it's because of this that we've been able to make a number of discoveries. So the first one that came out of this was, of all things, blue eyes in Siberian Huskies. So eye color is something that owners can easily tell in their dog. You can just ask a question to an owner: what color are your dog's eyes? And we did that as one of several questions that we asked owners. And not unexpectedly, we saw an association on chromosome 10. So I don't know how many of you have seen these kind of Manhattan plots before, but the idea is you get these kind of like skyscrapers where you have a significant effect. And so that's the region of the genome that has a variant that's associated with some condition or phenotype of interest. And so in this case, we have a small little blip here in chromosome 10. That's the Merle locus. We know that Merle dogs can have blue eyes or heterochromia, so it wasn't surprising that we had an association there. But on chromosome 18, there wasn't any known canine pigmentation gene in that region. We had no reason to expect that that would be associated with eye color. But sure enough, in the dogs with Siberian Husky ancestry, where the owners reported blue eyes, we saw this highly significant result. We looked at sequencing databases of Siberian Huskies versus other breeds, and we found many of the Siberian Huskies that had this SNP variant, which was on the array, also had this clear evidence of a duplication, a fairly large one, almost 100 kilobases long, upstream of this gene ALX4, which again hadn't been implicated in pigmentation things. And so we had this great association and this candidate mutation that we thought was probably driving the association. One of the great things about having people having tested their dog and loving new information about your dog is you have a ready list of individuals that you can turn to try to validate when you have an association like this. And so, of course, some people had told us what their dog's eye color was, but many people hadn't filled out that survey yet. And we could look at their genetic data and see, is their dog a Siberian Husky or mix? Does their dog have this variant or not? And can we infer whether or not the dog has that duplication or not, which we were able to do. And so you could send out a follow-up survey to people who hadn't told you anything about your dog's eye color. And we actually had then a very specific dog eye color survey where we had pictures and we showed the different kinds of dog eye color. And we sent it specifically to these individuals who had dogs with partial Siberian Husky ancestry, who hadn't told us about their dog's eye color. And we could tell whether it had the SNP marker and or the duplication that we thought was associated with it. And so sure enough, we could run that. And so our initial association was based on nearly 3,000 dogs, and very quickly we could do this follow-up study, and we got over 2,700 survey responses, and we could see how well the phenotype correlated with both of those. And we got a very strong association here with the SNP that was on the array originally that was driving the association, but we got an even stronger, much stronger association with that duplication. So it was really the duplication that was driving the association. It just wasn't something that happened to be linked to the SNP that was there. And so we could work out the genetics of this. So this is an autosomal dominant, not fully penetrant, partially penetrant mutation. And so dogs that have one or more copies of the gene are likely to have blue eyes or heterochromia. And they're more likely if they have two copies, but still one copy, it's enough that we at least partially dominant. And if the dog has the mutation but doesn't happen to carry the blue eyes, it happens to have brown eyes, it still passes the mutation in the blue-eyed phenotype on to its offspring. So it's a really interesting gene, and it's now incorporated as part of the test. So anybody buying a standard Embark test will get this blue-eyed result from this association. Another study that we published just last year, and this was work from Taki and others at Embark, is this for the phenotype of roaning. And so I think everybody is pretty familiar with this phenotype. It's in many setter breeds, as well as Australian cattle dogs. And it's also, something that owners can tell you about, or you can infer it just from the pictures that are uploaded to the website. Most people do upload a picture of their dog at some point, and that can be used by researchers to make these sorts of coat pattern inferences. It wasn't a surprise to us, of course, that roaning coat was going to be associated with mid-F. The spotting locus. So the mid-F is another white spotting locus. It's a very complicated genetic region, but it's sort of a necessary precursor. If you don't have the white spotting, you're not going to be able to modify that into a roaning phenotype. And but when you looked at the roan versus non-roan dogs, there was this very clear association on chromosome 38. And in fact, this is another case where the association was identified with a SNP that was on the array, but that SNP was linked to a duplication which was actually driving the phenotype. And in fact, the roaning phenotype also is dominant. So it only takes one copy of the duplication in order to drive the roaning phenotype. And another neat aspect, again, so we have these databases of the genetic data associated with the inferred ancestry from all of the dogs, as well as pictures for many of the dogs. And for those of you that really understand genetics and breeding, you understand that when you have a dominant gene, it's actually rather difficult to fix that gene in the gene pool, because dogs that are heterozygous for it still exhibit the phenotype. And so you're not able to prune out the last of the dogs carrying the recessive version, because when that recessive version is rare, it very rarely comes across two copies in a single individual. And so in fact, when we looked at the Australian cattle dog data we had, the dominant mutation, the roaning mutation, was at about 99% frequency. And so that meant only 1 in about 1,000 Australian cattle dogs would be expected not to carry at least one copy of that dominant gene. In fact, at the time, we had about 1,000 Australian cattle dogs in our data set. And in fact, one of the Australian cattle dogs in our data set did not carry any copies of the duplication, which we thought was interesting. And so, of course, the first thing the scientists did was take a look at the picture that the owners had uploaded for that dog. And when they did that, we could see here, this is Bodhi, and he's an Australian cattle dog that doesn't have any copies of the roaning locus. And so this is a pretty strong indication that the variant that we identified is the one that's driving the roaning phenotype, because it's segregating with that phenotype even within breeds where you don't traditionally see a non-roan phenotype. And so this is, again, part of the power of using what we call it community science, because it's our scientists working in concert with the owners and the breeders that are testing their dogs to generate both the genetic data and the phenotypic data that's needed to make these associations. Another neat coat pattern project that was done, this was done by another one of our scientists at Embark. This is from Dr. Andrew Slavny's work, is disentangling the intensity locus, the eye locus. This is something that's been known for quite some time in dogs and debated. And in fact, we see clearly that there's multiple eye loci. And in fact, she identified 5 different genomic regions on 5 different chromosomes that together can explain over 70% of the variation in pheomelanin intensity in the coat. And so for those who aren't familiar, maybe it's not important in your breed, this is the difference between sort of a cream-colored lab and a red lab. This is the variation that you see in golden retrievers coat intensity, but also in other dogs. Why are Irish setters very red, and why are other breeds much more cream-colored? And many of these breeds it's fixed, and in other breeds it varies through the whole gamut. And these associations are really interesting to me. So 2 of them had been previously identified, 3 are completely novel. Again, some research had been done on this before. One of the genes that we found was TYR, which is a fantastic gene. It was one that had been hypothesized as the eye locus, or at least a contributor to the eye locus in previous studies, but nobody had actually been able to prove an association, and that's because these were done through candidate gene approaches, where they said, oh, TYR is a good candidate, let's sequence all the exons and see if there's a difference. No, there's not a difference, so I guess that's not the gene that does it. But in fact, this is a non-coding mutation in TYR, so it's controlling the expression of the gene, but not the actual sequence of the protein that's underlying the phenotype. So even when you can guess the genes right, it's really, really useful to have dogs on a genome-wide array so that you can identify even non-coding variants, you can identify all of these modifier variants. And this research is continuing. I mean, we believe there's probably other variants out there that also contribute to pheomelanin intensity, and maybe they're important in other breeds. But we were very pleased to be able to build a model that basically doubles the amount of predictive accuracy versus what was already available. And again, it's just taking the genotype data that was already generated to do the genetic testing, and then taking the phenotypes from the pictures that were uploaded for the dogs and being able to do these things. The last study I want to talk about, which just came out a week ago, but it was a project that was a long time in coming, is early adult onset deafness in Rhodesian Ridgebacks. And so this was a project that started a decade ago, and before Embark was even around. And this was with Project Dog, Allison Rue, Mark Neff, and others working to collect samples from these dogs that were exhibiting early adult onset deafness. It's a recessive condition. I mean, you could work out the heritability. Project Dog did a fantastic job, raising funds, working with breeders, and getting to the point where they could find a locus on chromosome 18 that clearly was segregating with the phenotype, although not quite perfectly. There was not a causal variant that was identified, but instead they developed a linkage test that could be used to help inform breeding and help avoid the production of deaf offspring and identify carriers to improve the breeding. And so the project sat there, and again, linkage tests can be problematic because they're not always predictive. You can have recombinant animals, and so you really do want to figure out what the mutation is and offer mutation tests when at all possible. And so a couple years, 2 years ago, we were approached by the Ridgeback community, and they they wanted to get a better test out for their dogs, and it was a complicated effort. I mean, so the first thing is validating with our customer samples and other recruited samples that the association still holds, and it did. The deaf dogs did almost all carry that haplotype, and the hearing dogs almost always had at least one of the non-deaf haplotypes, at least in Ridgebacks. Although we could also, because we were testing all the dogs that came in and not just Ridgebacks, we had a very large database, and we could see that haplotype was also segregating in a few other breeds, including corgis. So about 10% of corgis seem to have the deaf Ridgeback haplotype, but of course the corgis weren't deaf. So that was one of the important insights that helped crack the riddle. And there was sequence data that had been generated. No candidate causal mutations had been identified, but our scientists looked more closely at the region. We saw there was a really good candidate gene in the region, epidermal growth factor receptor pathway substrate 8L2. This is a gene with a couple dozen exons, but it's in a really high GC content region of the genome. And so, what that means in non-technical terms is that when you do the sequencing, it's very difficult to map reads to this region of the genome. And if you don't have reads that are mapping to this region of the genome, you're not going to identify variants, including potentially causal variants for the phenotype. And so that meant our scientists in the wet lab, and so Vandana did a ton of work along with Taki, sequencing through each of the known exons for the gene to identify whether there are any genetic variants in this gene that could be causal. And they found a very good mutation in one of the exons of the gene. Boom, this deaf dog down here that was segregating with the haplotype had a 12 base pair deletion. So this is a deletion of 4 amino acids in the protein. They did a lot of modeling work looking to see, was this going to change the structure of the protein, and indeed it would. And then we could go back and we could sequence through a lot of other dogs from other breeds that also carried the haplotype that had been associated with deafness, but weren't deaf at all. And sure enough, when you sequence those dogs from the other breeds, they don't carry this deletion. So it's that deletion, which is linked to the haplotype in Ridgeback, is what causes the deafness, and that haplotype, when it is not linked to the deletion, which is the case in other breeds, doesn't cause the deafness. So again, having these large databases, these diverse databases, and this phenotypic information from the owners and breeders is really the key for unlocking it. And so I think that sets the stage for the way we think about the future of genetic testing and how it can improve dog health. And so the great thing is we had this data now from other breeds that have dogs that carried the haplotype that was associated with deafness in Ridgeback, but didn't exhibit the deafness phenotype. And sure enough, when we sequenced those dogs, we did not find the 12 base pair deletion. So we were able to, in Rhodesian Ridgebacks, this 12 base pair deletion almost always occurs with that haplotype, whereas in other breeds it's uncoupled. And we can see now then it's definitely that deletion that's driving the phenotype in Rhodesian Ridgebacks. And so now we have a test on our array, and dogs are tested to see whether or not they carry that deletion, and that's the basis now of breeding decisions. It's a much more powerful test, and it's specific for Rhodesian Ridgebacks. So with that now, I'd like to transition into talking a bit more about how we see the current state of genetic testing, the future of genetic testing, and how all of these discoveries and our ability to expand upon them is going to improve the long-term health and welfare of dogs. To me, it's a three-pronged approach. One of them is it's so important to do genetic screening of particularly breeding animals, but also for just your regular pet owner. There's so much that's known about dogs now, and it's relatively inexpensive compared to other kinds of preventative and diagnostic care that you give your dog, that you ought to do this screening just to make sure you aren't propagating the potential for affected individuals, or if your dog is affected, that ahead of time, or has a genetic propensity to be affected, that ahead of time, and you can take action to help improve your dog's health. So this is a picture of Wolfie. This is one dog the owner tested with us, not a breeder, shelter adopted dog, wanted to know which breeds were in the mix, however, also got the health report and found out that Wolfie was at risk for hemophilia, which is something they would have never known. And sure enough, months later, Wolfie was in an accident, and because the owner knew that the dog was predisposed to hemophilia, took the dog immediately to a specialty clinic where Wolfie could get the blood transfusions that he needed, and he survived the accident. And we get this very happy letter, "You saved my dog's life," which was, it's always a warm, fuzzy feeling when we get those letters. I'm proud to say that when we survey pet owners who take Embark and get the full panel done, 61% of them, change their dog's routine or feel better about, the care that they're giving their dog based on that genetic profile. And we've also given the test to veterinarians and said, hey, give this test to your clients and let us know what happens. And when we do that, we get very similar, results. So in this vet clinic, they tested 100 dogs. 27% of them were clear for everything that we tested for, which is about what we see in our overall population. 25 of them were carriers, which doesn't change the care of the dog, but certainly would impact breeding decisions. You'd want to make sure you do a carrier screen of any potential mate there. 22% did have risk factors for various diseases, and the vets reported 3 of them actually had drastic changes in care. The lives of the dogs were saved, from this, and so it could be situations like Wolfie or other, kind of highly impactful diseases that are treatable and preventable, and to have this information, or even like drug sensitivity, things like that. So we definitely think this, genome-wide panel testing is important as one of the prongs for improving dog welfare and health. I think the second prong, though, is thinking more expansively about what genetic testing can do, right? We now have a database with 1 million dogs. And if we can connect that database to phenotype data from 1 million dogs, we now have enough data that we can start to develop things called polygenic risk scores and genetic breeding values. And we can, there's not a cancer test for dogs, right? I can't say whether your dog's going to get cancer or not. And in fact, cancer is a very complicated, phenotype. The genetics of it are going to be very complicated. But we know that it's heritable. We know that different breeds get different cancers. We know it's the #1 cause of death in adult dogs. And so we know that if we get enough cancer diagnoses and we have enough genetic data, we'll be able to start to predict which dogs are at highest risk of certain cancers, which may change the way those dogs are screened. It may also change breeding practices, right? A breeder can decide, cancer is really important in my breed, and so I want to make sure I don't breed dogs that are going to pass on high probabilities of cancer. Or maybe they'll decide that heart disease is more important. You have to pick and choose what you're breeding for. You can't breed for everything all at once. But we want to give breeders the tools and get the data so that we can give them the tools so that they can make these sorts of breeding decisions. So the way we look at our database right now is our main tool for getting these phenotypes is the annual health survey, right? And I'm proud to say we got over 200,000 owners and breeders to fill out this survey last year. And so it goes, from the very beginning, just rate your dog's health on a scale from 1 to 5, into, more complicated, different organ systems and different diseases. And that data is going to be key for driving discovery in the future. And the way we look at it is most people who test their dog with us are testing puppies or newly adopted dogs that are young. And of course, most of the diseases of interest are things that happen in middle age and older dogs. And so if people keep coming back and keep telling us and updating us about their dog's health, we're going to be amassing more and more samples over time that wound up affected with different diseases or clear of different diseases. And we can build the genetic models to predict which dogs are going to wind up in the affected or the clear classes and what that probability is. And then develop these breeding scores so that we can reduce the prevalence of whatever the key disorders are for different breeds. And I think that's a really important aspect for improving dog health. We do have these polygenic models already on our website for things like genetic weight, and so we can build similar models once we get the data for these disease conditions too. And so that's what we're looking for. And then the final thing I want to talk about is genetic diversity, and particularly how it relates to inbreeding. Different dog breeds have different levels of inbreeding, and there's lots of variation within a breed between the dogs that are the 10% most inbred and the dogs that are the 10% least inbred. And so on average, Doberman Pinschers and Bulldogs are more inbred than Poodles and Beagles. But of course, you can still get inbred Poodles and you can still get inbred Beagles. And when I'm talking about inbreeding, I think a lot of people don't really have a picture in their mind of what inbreeding looks like. And what it looks like to a geneticist is these dogs have 38 autosomes, and the inbreeding are these tracks where large sections of chromosome, the one that they inherited from mom and the one that they inherited from dad, are identical. So in this case, it's painted blue, right? And so when you have that, if that stretch of chromosome harbors a deleterious recessive, that means you got 2 copies of that deleterious recessive, and the dog's going to be affected by that. And of course, we can test for the known 200 plus recessive mutations that have been identified in dogs, but there's thousands, if not tens of thousands, of deleterious recessive or partially recessive mutations in dogs and any organism. So the vast majority of this we can't test for, but we can tell you, your risk is going to be proportional to the amount of what we call identity by descent that you have in the genome. And so we do this, again, on a genome-wide scale. So each one of these chromosomes has thousands of markers on it, and so we're very easily able to see, oh, here's a stretch of 500 markers that are all homozygous, right? This is an identical by descent stretch that's caused by inbreeding. And so it's very different than kind of these STR-based approaches where you're only looking at a couple dozen regions of the genome. You don't even have a marker on every single chromosome. So you're not able to either the STR falls in a region of inbreeding or it doesn't, but it's really just a by chance thing, and it's not going to give you that detailed picture about it. And in fact, we can overlay for different breeds where the regions of inbreeding happen, and gives you this you can kind of see at a glance. Oh, in Labrador Retrievers, we see most of the genome is outbred in most dogs. There's a few areas that look like, there's an uptick in inbreeding, but for the most part, Labrador Retrievers look pretty healthy in terms of genetic health. When you go to a Dalmatian genome, we actually see there are portions of the genome that have low or no diversity, and I've actually circled 3 regions of the genome that have no diversity in Dalmatians. And the first one is the mid-F locus, the white spotting locus. The second one is the roaning locus that I talked about earlier. So Dalmatian spots are at the root a roan spot, and then on top of that, there's this chromosome 3 spot modifier, which is also dominant. That leads to the Dalmatian spotting. And actually, this chromosome 3 locus is linked to the HUU locus, and so that's so you get these positively selected things, but you can sometimes have negative phenotypic health consequences from that if there's something linked to it. And then Dobermans, there lots of the genome relatively has little diversity or no diversity, and I would say for Dobermans, designing ways to keep the diversity you have is going to be really crucial. And and when we take this inbreeding data and we actually link it to the annual health survey data, we get some really interesting correlations. So if you take dogs that are in the top 20% of their breed for inbreeding and compare them to dogs that are in the bottom 20% of inbreeding for their breed, you actually see a pretty big difference in lifespan, 2 or 3 years, right? And so of course, lifespan is going to depend on what the size of the dog is, but it turns out that outbred big dogs live just about as long as inbred small dogs do. So inbreeding plays almost as big of a role in overall longevity as body size does. And not only that, if you look at dogs that were rated an excellent versus not excellent health, you see you're much more likely not to be in excellent health if you're inbred versus outbred. So these dogs are living shorter lives on average, and on average they're having more health issues. And we actually collaborated with Morris Animal Foundation earlier, and we could see that at least within golden retrievers, the inbred females were also having smaller litter sizes. So this is all, these are 3 different pieces of evidence showing that at the range of inbreeding coefficients that we're seeing today, that breeders are doing, we are seeing effects of inbreeding depression. So all else being equal, we should be, trying to breed away from high COI litters, and to the extent that we can replace them with lower COI litters, and we're going to get definite improvements in dog health today by doing that. So that's a kind of whirlwind for some of the data and some of the projects that we're working on here at Embark. There were tons of people involved in some of the initial studies in my lab and elsewhere at Cornell, and of course tons of researchers at Embark that have contributed scientifically to this, not to mention all of the engineers and customer service and product and everybody that has played a crucial role in enabling this really unique discovery system and building it out so that with the support of breeders and pet owners like you, we're actually going to be able to continue to drive discovery for more and greater important diseases and build the tools needed so that we can actually make a huge difference in the future of dog health. So thank you very much.
All right, and we are live with Dr. Adam Boyco. Thank you very much for that informative presentation. It was really interesting to me. I learned something new every time I hear you speak.
Thanks, John. It was a lot of fun.
Great, that's great. So there have been a number of questions coming in from the audience, and I see they're continuing to do so. So we'll get through as many of them as we can. As a general reminder, as I've been saying throughout the day, if we don't answer the question you've submitted, all of our attendees can go over to the Embark booth and post the questions there, and a number of our staff are available to respond via text as well. But we'll get through as many as we can here. And just to start it off, Dr. Boyco, kind of before we get into the technical aspect of a lot of these topics, could you just share a little bit what got you into this field of research in the first place, and why are you so passionate about canine genetics and health research?
I mean, I'm a scientist who's always loved dogs. I got my genetics training in a lab doing mostly human genetics work, and I was fortunate enough that they started a collaborative project on dog genetics. And getting to work on that project, it really crystallized what a great model dogs are for so many different genetic conditions that are of broad interest, not just to dog owners and veterinarians, but also to humans and human medicine. And so being in on that project and then sort of seeing not only the promise, but also, man, if we could just get large data sets of dog genetic data, what kinds of cool discoveries would we be able to make? And how can we, improve the lives of dogs today with the kinds of genetic information that you're able to produce? I mean, I just was super excited about that, and having the support of Cornell and NSF and other funding organizations to pursue that research and eventually get a faculty position and eventually be able to launch something like Embark has been extremely fulfilling and terrific to get this gigantic database that we can now use and collaborate with scientists to determine all of these new discoveries.
That's great. That's great. Well, a lot of amazing discoveries have obviously been happening in the last few years, and we're excited for what's coming next. So to jump into some of these questions, there's one, might be a good one to start with. It says, I just left a panel about COI with the message, we cannot study what we do not test. Is there a chance for whole exome sequencing in the future for greater coverage, or does the homogeneity in dogs from inbreeding mean very little is gained through this increased data?
That's a great question. I have worked with exome sequencing data before. It seems to be particularly useful in the context of cancer and understanding somatic driver mutations that different cancers have. The great thing about dogs and the homozygosity and linkage disequilibrium, that they have is that it does make it a little bit easier to do these initial genetic characterizations. And generally, 200,000 markers within a breed is going to be sufficient to accurately tag causal variants, even ones that aren't on the array. So you don't really get that tagging when you're looking at a few dozen sites in the genome, like with the microsatellite. Some of them don't even have any markers on certain chromosomes, so that would certainly be a problem for this kind of work. But when you have that many markers, the additional information you get from sequencing is a lot less. There's a place for sequencing, but we generally use it in the fine mapping portion. And you could do exome sequencing. The problem there is you're only looking at the exons, and we see a lot of cases where it's regulatory variation that's driving the phenotype. So we've seen this in blue eyes, we've seen this in the TYR locus for the intensity locus. And even when it is an exon, a lot of times it's a complex structural variant, maybe an inversion that's perhaps not well picked up by exome sequencing because the breakpoints aren't going to be there. So it's a very complicated question, and there's places for lots of different techniques, but having this genomic database for your breed based on really dense research grade genotyping data seems to be a very productive path towards discovery.
Got it. So a couple of questions about, is Embark researching something currently or planning to do so in the near future? A couple examples are canine rage syndrome genetics and the genetics behind different variants of brown eye colors.
Yeah, those are great questions. One of the early projects at Cornell that I had some visibility in, that the Biobank was collaborating on, was Springer Spaniel rage syndrome. And there was actually over 100 samples that were collected in that project, but unfortunately the genetics didn't seem as simple as what we had thought they would be initially, and so we never did get a really good locus to pop out from that. I don't know if there's other breeds that are characterized by these rage syndromes. I'd be really interested to find out. It might be an opportunity for crossbreed mapping. In terms of eye color, we found owner-reported, blue versus brown eye color was a lot easier for them to do than to try to discern different shades of brown. So we haven't really pursued that yet, although we do have an eye color survey that is asking, that question. We are collecting data on that. So So that is one of the things that we can look at in the future and see if there's something besides the known coat color genes that shake out for in some of these breeds for eye color.
So, related to this topic, we often get questions from both breeders and veterinarians about what, how does Embark sort of think about which research to engage in? And then how can people support it? You just mentioned a survey is why I thought of this. If people are interested in recommending to Embark, I'd love it if you research something, and or how can I help any research you have going now? Like what would you advise?
I mean, the first step, I think, is if you're testing with Embark, fill out the annual health survey for the dogs that you're testing, because that gives our researchers the ability to quantify how important a condition is in a certain breed. If we aren't seeing dogs being tested that have that condition in that breed, that's not really something that we're going to be interested in researching. But if we see that a large proportion of dogs from a breed are reporting this condition, so we already have some genetic data there, that gives us the ability, let's focus on this for follow-up. Do we need clinical information? Do we need to recruit a few more dogs? Do we want to look at cross breeds or mixed breeds and those kinds of questions? But without kind of having that initial information to be able to make an assessment about whether something is important enough and whether the samples already exist that will be available for study, it's really hard to move forward on a research project.
Got it. So a couple other questions here about ongoing research. First, is there any research into, is it ADDL or additional tall genes in Belgian sheepdogs? I'm not sure if that's the name of the gene.
I mean, we are doing a lot of size research. We've focused on weight because that's a little bit easier for owners to report, and it also relates a little more closely to obesity, and which is obviously of importance to a lot of owners and for dog health. But it's also true that there are some breeds where we do, breed dogs where we are routinely underestimating the body size, more than typical. So there are size variants out there that are unknown to science and be a really interesting area to follow up on, and particularly if there's breeds that are segregating for these relatively large effect height mutations, that it's great to know and see if we can design a project around that to disentangle it.
Got it. And then one other one, testing for fluffy or undercoat.
Yeah, so I'm not aware of any published research around that variant. It's something that we will monitor. I mean, if there is a mutation out there that seems causal and worth testing for, we would certainly consider adding it to our panel. But right now, we haven't really seen any, scientific work kind of demonstrating a causal variant for that locus.
Got it. So, so many questions are continuing to come in, and I see a couple of themes around COI, inbreeding, and then ancestry related. So, starting with COI, kind of combining two questions here. Is there an ideal COI, and does that depend on breed?
Right. So the short answer is no, there's not an ideal COI. It's going to differ for different breeds, and COI isn't like conformation where the dog that is best in the show ring wins a blue ribbon. It's either it's a probability and it's a degree to which the potential to suffer from inbreeding depression exists. And so, how much risk are you willing to take on that? And it's going to depend on the lines. It's going to depend on what the background distribution of COI is for a breed. The way inbreeding is computed, it just means that two sections of a chromosome are identical by descent. So it's because they came from the same ancestor. There was, inbreeding back in the line somewhere. Well, some of that could be fixation of beneficial traits, right? You don't want to breed that out. And so you kind of have this like minimum COI to be like well conformed for the breed, but then you also have these additional stretches that really aren't necessary for conformation, and to the degree to which they harbor recessive deleterious variation, and we've seen in numerous different data sets and numerous ways of studying those data sets, that in all of these breeds they are harboring, some of them are harboring recessive deleterious variation. You just want to reduce your exposure to that sort of health risk. And so all else being equal, a lower COI, towards the, bottom half of what you're seeing for the breed is going to be desirable.
Got it. So related to COI, Embark has participated in research across a couple of different areas. One, accurately measuring inbreeding with like the Doberman studies, and then inbreeding depression in golden retrievers. We participated in that study with the Morris Animal Foundation. And that, those two, I mentioned those because there's a question here that says, what is the proposed mechanism behind the effects of increased COI on lower litter size and life expectancy? So I wonder if you could kind of talk about that topic overall.
Yeah, inbreeding depression is, seen throughout the animal and plant kingdoms whenever you have inbreeding in populations, and dogs are no exception to that, as we're seeing now with the ability to accurately measure degrees of inbreeding using genomic data, and then the ability to tap into these data sets of well-genotyped dogs for which you can accurately calculate inbreeding and see what's the longevity of the dog, what sorts of health predispositions does the dog have, what is the breeding history of the dog. We see in all of these measures evidence of inbreeding depression from dogs that are on the higher side of inbreeding versus the lower side of inbreeding. And so, again, it's because everybody in their genome harbors what we call deleterious variants. And these are things that, reduce the effectiveness of a gene or maybe knock it out entirely, which doesn't matter if you only have one copy, but if you have two copies of it, then it causes health problems. And maybe there's some clinical health problems, but they still impact growth, they still impact fecundity, they still impact longevity and health. And so the more of your genome that is identical by descent, where you know that both copies are going to be identical of whatever you inherit, the greater your probability of having inbreeding depression and the problems associated with that.
Got it. So I know this gets a little bit away from research and more towards just kind of applied breeding decisions. But there's a question here that we also saw in some of the earlier presentations about, I think, basically balancing the kind of short-term and long-term. So there's a question here that says, how many generations do you recommend waiting to perform a line breeding in order to try to preserve positive genetic traits in the line? And what came up in some prior sessions is essentially, how do breeders balance these two competing forces, where I want to have particular phenotypes, in my dogs, and I might have a great breeding candidate based on other attributes and temperament, but the COI, isn't ideal, and the projected COI of the litter isn't as good as it could be with a different dog. So how do I balance these two kind of sets of priorities?
Right. Well, how does a breeder balance any set of priorities, right? They have to choose what's important to them and their line. And, breeding a dog that comes from a line where, maybe cancer's been reported in several of the individuals, but you're really trying to breed for cardiac health because that's the biggest problem that you're having, right? Like, maybe that's okay. But in other cases, maybe you don't want to do that, because cancer is the biggest problem that you're seeing in the lines, and it's not worth it to increase that risk just to fix a certain spotting pattern or something like that. It really is the decision of the breeder, with as much information as possible, like, what are the health problems, what are the outcomes I'm going for, and recognizing that this is one aspect that I should manage among many. You would never recommend that a breeder crosses two carriers just to, for a bad early onset health condition, just because that's going to lead to a lower COI than a carrier clear cross, right?
Right.
So COI is not the only determinant that you want to use, to make these decisions. And likewise, two dogs can be compatible for all the genetic tests that we do and have a relatively low COI, but if they don't pass health testing, if they don't have good, hip conformation or whatever the main issues are for the breed, you still don't want to breed them, right? So genetic testing is one component of many of the decisions that breeders need to make. And, frankly, if you don't select for health and genetic health, you're not going to get it, right? That's the way select. Just like if you don't select for temperament or for conformation, you're not going to get it. Like, eventually this all decays. It doesn't like run on autopilot. natural selection works generation after generation, and if artificial selection is not doing the same thing, then you're not going to keep it in your line.
Got it. Got it. All right. So one other question related to genetic diversity. We only have a few minutes left here for questions, and then we'll close with a few other quick topics. But this question relates to something I know you're fairly passionate about. It's asking about, is there a way to check MCH class 2 DLA, DRB1, DQA1, DQB1 data, and then to see if there's a connection between allergies and dogs, that have lower diversity or no diversity. I know allergy testing is something you're interested in for the future.
Right. So we have not observed that kind of relationship between allergies and MHC. With the array-based testing, we don't produce MHC haplotypes according to the microsatellite nomenclature, and so for right now we're not doing that sort of information. And also the phasing of the region is a little bit more difficult too, so we want to make sure that we get the science right before we launch something like that. I mean, undoubtedly MHC is an important region of the genome, although ultimately it's a very small part of the genome, and we clearly are seeing effects of genome-wide COI on health outcomes in a way that we're not so far seeing similar magnitudes of effects with MHC in particular on these phenotypes. That's not to say that the research tomorrow isn't going to show something else for us.
Great. So a question here on mixed breed ancestry that I'm sure a lot of people have asked about from time to time, which is, essentially how is this done, and how is it that Embark is accurate in saying, a dog might only have 5% of a particular breed when, 95% of the dog is, one or two other breeds?
Yeah, so I mean, we run some pretty sophisticated statistical algorithms on a really large reference panel of dogs of known breed ancestry, registered dogs, etc. And so, we know when we have a mixed breed dog where the amount of shared ancestry diverges from what the expectation would be in let's say a two-way mix. And so clearly there's some more ancestry coming in from third breed here. I mean, it's really a bottom-up approach where we're finding shared segments that kind of indicate relative, history, and then figuring out, okay, well, so then looking at those segments and adding them up, how much of the genome is coming from that breed versus the other breeds.
Great, that's great. All right, so then just a last couple of questions here that I think probably relate to your earlier presentation and your overall work at Embark. But basically, if you look back to, I've heard you speak about your original vision for Embark and how Embark's products would generate data that is, or that facilitates research. Could you just highlight a little bit more about what you mean by that, and what are the kind of innovations or discoveries you see on the horizon coming soon that you're excited about?
Yeah, so really excited to be, at the stage now where our scientists are making discoveries that we're able to put back on the testing array, and so continually improving the amount of information you can get from dog genetic testing, with just more tests, but also working towards different in-kind tests where we're like explicitly accounting for complex risk factors and giving breeders the opportunity to breed away from some complex conditions, but instead of just doing carrier testing, which is primarily what's going on right now. Also, like the ability to, launching a veterinary product which enables genetic testing to happen in the clinic and get associated with clinical data, I think that really increases the rate of discovery, which I'm extremely excited about, as is the new age test, where we're launching a product now looking at methylation marks, which do change over time during a dog's lifetime, and therefore can be used to infer what a dog's age is. So this is probably less important to breeders that already have exquisite knowledge about all of this information for their dogs, but the methylation data in and of itself, I think, is useful to the scientists and eventually will be useful in different in-kind tests, looking at various aspects of dog biology and risk factors and things that maybe are not really readily testable for just using genomic data, but epigenetic data can be extremely informative for, perhaps things like nutrition and how well a dog is tolerating a diet, for instance.
That's great. That's great. Yeah, I'm excited about a lot of these items coming. Obviously, working at Embark, I get kind of some insights into the work going on. But I think a lot of people are really looking forward to the work you're talking about.
Great.
So as a last item, do you have any is there a key message or like a call to action or kind of like a final takeaway you want to leave people with before we wrap up?
Yeah, I'll sound like a broken record, but for those of you testing with us, please fill out the annual health survey. We really use that information to then help figure out what kinds of discoveries should we be focused on in the next year, and what kinds of outreach, and certainly breed clubs that have high rates of filling out those things are darlings to us, and we want to give them as much love as possible in our future research efforts.
Fantastic, fantastic. All right, well, thank you again very much for your presentation and participation here in the Canine Health Summit. Again, Dr. Adam Boyco is Embark's Chief Science Officer, we really appreciate the time you spent here. And now looking forward for all of our attendees, we have a bit of a break until 3:45 p.m. Eastern, when our next presentations will kick off. So in the meantime, you're welcome to continue exploring the Summit website, the conference or event website. A number of tools and features available to you, including chatting with other attendees and colleagues, as well as asking additional questions to the Embark team that we may not have had a chance to answer here live on the air with Dr. Boyko. So thank you again for everyone's participation, and we will see you back in a little bit.
Thanks, everyone. Thanks, John.